this is Medicine in 3 Minutes, and today we're going to be talking about the blood-brain barrier. So in 1885, Dr. Paul something, sorry I can't pronounce his last name here, decided to inject trypan blue in a mouse. And when he did so, he noticed that the body and its systems were all stained in blue, except for the central nervous system and the peripheral nerves that usually would stick out right here. And obviously that raised the question of why didn't the coloring agents stain the central nervous system and the peripheral nervous system? And that obviously led us to the blood-brain barrier. So a quick little definition is that it's the selective semi-permeable membrane barrier that separates the circulating blood from the brain. So semi-permeable obviously means that it allows only a few things in. So through passive diffusion, water and liposoluble molecules can get in, no problem. It does not let in lipophilic neurotoxins, and via selective transport, glucose and amino acids can find their way in. And when we say selective is that they must meet certain criteria. So functions of the blood-brain barrier is that it stabilizes the microenvironment of the neuron. It protects the neurotoxins to enter the central nervous system. It maintains neurotransmitters within the central nervous system. So neurotransmitters, think dopamine, acetylcholine, serotonin, etc. It mostly wants them inside the central nervous system as opposed to roaming free in the body. And it prevents free movements of chemicals from the blood circulation to enter the central nervous system. So in order to understand how the blood-brain barrier works, we have to understand how the capillaries function and their anatomy.